We have lots of stuff to talk about with enzymes, and not a lot of time to do it. All right, so here we go. Last time we were together, we mentioned this idea of there are two main types of reactions that happen in living cells, right? There are anabolic and catabolic reactions. Right? Anna the builder, right? So anabolic processes or reactions are going to take small things and build big things. So they're taken in because we're loading energy into that larger polymer or molecule that we are now constructing. Yep. Energy is going in, so it's ender. Got it. Good. Okay. With catabolic reactions, it's much the opposite. We're taking big things and breaking them into smaller things. It's released, absolutely. So would that mean hit exer or ender? Got it. Yeah, go ahead again.
Exer, got it.
Yep, you got it. Yep, absolutely. So, and that I would also include the umbrella if we were measuring temperature, possibly seeing it being exothermic, giving energy out, heat energy out, possibly. All right, good. See, so we're in good shape. And so we should notice that a cracker, a starchy cracker, if we munch on it, will go from tasting like the salty coating that they put on it and the flavor they add to actually like kind of a sweet taste underneath all of it. And that's due to the fact that we have this amylase enzyme that's going to break down starch 1,000,000 times faster than it could be done alone. Right? And so that's where we're going to focus our attention on is like, what are these special organic catalysts, these enzymes? What do they look like and how do they work? We're going to look at that. So how do they actually help? This graphic is something that we'll look at breaking down in just a moment. But remember that inside of the human body, like we're locked into that 98.6 degrees for our temperature. So we can't really provide more heat energy to help the reaction happen. If we do, we start to ruin the proteins that already make us up. We need to make use of a catalyst, and in this case an organic catalyst, that will allow us to perform the same chemical process but require less energy to activate it. And so that's what these enzymes are going to do. They're going to reduce the energy needed for activation. And that activation energy is simply energy needed to start a chemical reaction, or start a process. So if we're given a graphic like this, we're kind of looking at, how does an enzyme change the activation energy of the system? Right? So I highlighted the first higher graph or plot, and we can make note of several things. One, the energy of the reactants at the beginning it will be at a certain level, right? So if we consider that starchy cracker that we're munching on, right, to break that down into simpler sugars, that's going to take a certain amount of energy to happen, but we know that the starch, the cracker we're munching on, has a certain amount of energy about it, right? That has not been changed. And we provide it with a certain amount of energy to reach activation, right? And that's quite a bit. It's a very rather steep hill for us to climb with energy to get this reaction to start the fire off in the first place. The reaction happens, and we produce the products at the end. We produce those simple sugars. We break this very long starchy chain down into more simple sugars. Right? And they're going to have a certain amount of energy associated with them. Looking at the shape of this red plot, what type of reaction is it? Is it catabolic or anabolic? You got it, yeah. So from beginning to end, we've lost energy. The system, the products have less energy than what we had to begin with. That's energy out. So that probably suggests that we're going from larger things to smaller things, and we're also getting energy out in the process. Sure, absolutely. Right now, this is requiring a good amount of energy to activate. So if we switch our focus to the other plot, when we actually use an enzyme, right? We line that guy in green. Notice that the energy of reactants hasn't changed, right? We're still munching the same cracker, but now we have this enzyme amylase that we're going to use to help this process along. And so what does that do? Well, that reduces the amount of activation energy that we need. Right? It allows us this reaction to happen with less energy, and if that's the case, it allows it to happen even more quickly. So we kind of make a note of that the activation energy used with the enzyme is less. And then notice that our energy of products at the end, we still get to the same end result. So whether we use an enzyme or not, the reaction that we're looking at is still the same. We still have the same reactants, we still have the same products. The difference with no enzyme to with an enzyme is how much energy did it take to get it started in the first place. Is the reaction still catabolic? Yes. We still end up with less energy in the products at the end. We still end up with the same types of products at the end, whatever this reaction might be. It's just that the energy taken to get it going is reduced. This is a similar plot, but is this a catabolic or anabolic reaction? All right, Anna the Builder. Anabolic, yeah, you're absolutely correct. But you need to be able to look at this and kind of decide that, right? Now, you're right. The dotted line, we don't go up as high because the enzyme's helping not need as much energy. Right? So we can still end up with the same products. They have the same amount of energy stored. The difference is still the same from beginning to end. It just didn't take us as big of a hill to climb over to get that to happen. But the reason I want to show you both situations is because I don't want you locked into the idea that enzymes only work one way. Right? We have a multitude of enzymes that help us build things and a multitude of things of enzymes that help us break things down too. All right, so in case you need like a better analogy, it's like trying to shove a boulder over a steep hill. Okay? The reactants before is the boulder at the beginning. We reach activation energy. The reaction is rolling down the hill, and then stops at a certain point when it levels out. All right, so this would be without our catalyst. The catalyst or enzyme, which is going to be a biological catalyst, right? An enzyme is going to reduce the steepness of that hill. Enzymes reduce the amount of energy needed. So if you do not use enzymes, you need more energy to make the same process happen. This is sucrose, a disaccharide, and we want to break this sucrose down into the monosaccharides that make it up. Right? It's going to require hydrolysis to make this happen, right? And so to go from a larger thing to a smaller thing, this is catabolic. But this reaction to break sucrose or sugar down into its simple bits, it takes a lot of heat energy, a lot of energy in to make it happen. So we have to crank up our hot plate or crank up our Bunsen burner to help this along. And then two, it also has to be in a very acidic environment to help try to coax these guys apart. Right? That's without enzymes. Sucrose will use a special enzyme. Sucrase will break down sucrose. And so this reaction can happen more quickly and in a neutral pH, so that's awesome, at body temperature. So how does the enzyme do this? So let's look at the structure of an enzyme. Okay, so let's just consider that this big dark gray blob is our enzyme. All right, so this is, we can even use this with our first example, with amylase breaking down starch from a cracker we're chewing on. All right, so this is our starchy cracker. This is our known as a substrate. And we have our enzyme, our amylase. Okay, so the specific shape of that the enzyme makes, where the substrate would actually fit in, is known as the active site. So these active sites on enzymes are where they're going to do their help to make these reactions happen. And they have a very specific shape. Like it has to be like it's intended for a specific type of substance. Right, in this case, this enzyme is amylase. It's built specifically to break down starches, long polysaccharides. The reactants are also called the substrates. What's being worked on and changed. And so with our amylase and starch example, starch is a substrate, amylase is the enzyme. So this enzyme's hanging out there, has this active site open, and starch floats nearby, and it gets attracted to the active site of this enzyme, and they bind together. And if it fits, then the enzyme will actually kind of grip or hang on to this substrate that is now cozied up into its active site, if it's the right thing. It's like a lock into a key. Like in the school building, like I have a key for my room, so if I take that key to the athletic complex, it will not like open like certain athletic storage locks. The key will fit in, but I can't turn it. Right? So there might be things that float in and fit, but they can't turn the lock. So they just move on their way. Like nothing happens. And then says, sorry, bud, I can't work on you. I'm not meant for you. Right? But in this case, we have amylase working on starch. So starch cozies up, it wraps around it, and then now, depending on what it wants to do with it, if we're trying to break it apart, the enzyme will help lower energy needed by tugging on what makes it up. It'll start to pull apart or try to gently tug away the bonds that make up this longer item we're trying to break down. If we're trying to build them together, we're trying to make a longer chain of things, then it'll squish them together. It'll try to force them closer together, because in chemistry, in biochemistry, if we want a reaction to happen, it's all about collisions. Okay, so the enzyme is there to kind of get them together, but it's not going to participate in the party. All right, so these guys get either pulled apart or shoved closer together, and then the chemical reaction can take place between them. And when it's done, we're going to now have products. So when they first get connected, there's still the substrate with the enzyme. So this is an enzyme-substrate complex. After the chemical reaction happens, since we've lowered that energy needed to do it, it's now known as the enzyme products complex. So it's enzyme because of the enzyme, substrate, and complex just mean they're together. We'll temporarily rename them as the enzyme products complex, because the enzyme has done its job, the reaction has happened, and now we have products hanging out in that active site. All right, once that happens, the enzyme knows that these guys have been changed, because they no longer have the desire or connectedness to hang out in that active site anymore. So the enzyme opens up and releases them, and they go on their merry way. Maybe they're going to be used for something later on by another enzyme, or maybe they've reached their final product. It kind of depends on the process. But the key thing to keep in mind through the whole thing is that the enzyme does not get changed. Right? The enzyme can be used again at the end for the exact same process all over again. We can also have enzymes that put things together. Right? And the same idea happens all over again, where we have two smaller bits, and then enzyme C is going to have certain shapes to it where these two bits can fit in close to one another. And then it's going to kind of cozy up around them and force them closer together. The reaction doesn't require much energy anymore. It happens, and these guys get bonded up and released from the enzyme, and the enzyme's ready to go again. This is like a string of glucose, like a starchy sugar, okay? And this is amylose. We're going to use amylase to break it down into 2 disaccharides. I'm going to simplify this just by saying we have a string of glucose kind of linked together, all right? And we're going to use a specific enzyme to break these apart. Amylase says, oh, dude, I can work with that. So it cozies up, binds with this guy, and then they are placed into the active site of amylase. Amylase helps this reaction happen, and they get cleaved apart. Right? We form a couple disaccharides out of this. Sometimes students will ask, well, why doesn't amylase just do this again? Because it can separate one glucose molecule from another, but you have to remember that this was once a very long chain, or there could be a longer end to the other side of this. And that's what amylase works on, specifically cleaving larger starches. It's not built to do smaller things. We need something else to work on the smaller things. So if we encounter a similar setup, we can take that amylase enzyme that got done working on the previous strand of starch, and we can move him over, and he'll work on the next one for us and do the exact same thing again for us. But these enzymes are specific and reusable. We can use them, but for the same job, and that's it. We can't give them new jobs. We can summarize the process into a nice cycle. Step 1, we have our substrates enter the active site of an enzyme. Step 2, they'll bind together, and this enzyme-substrate complex forms. Step 3, now that we're inside that enzyme-substrate complex, that chemical bonds, if we're trying to break them apart, we're going to start pulling on, it's going to start tugging on those bonds to try to separate what's there. We're trying to build things and put things together, that's going to try to shove them closer together. And that's going to require then less energy to do the next step and actually have the reaction occur. It'll take less energy to do that. Step 4, our substrates then are finally converted into their final product, either breaking them apart into smaller bits or merging them together into a larger bonded molecule. And at that point, the enzyme knows the job's done. I do not need to hang on to these anymore, and because they've been changed, they no longer kind of link and bond with the enzyme as they once did. So they get released from the enzyme in step 5, and then in step 6, the enzyme kind of gets reset and is ready for the next round of substrate to come in and be worked on. So I thought it would be kind of interesting to also include some information about the active sites maybe. So I found a couple images that worked really well. This active site is very specific. Right? It's the part of the enzyme that attaches to that substrate, and the shape of each enzyme is absolutely very, very specific to the type of substrate you want to work on. Right? It's like a fitted glove. It's like taking your hand and we get like a 3D mold of it, right? And then we hand sew a glove around that model of your hand, and so now you have a glove that fits perfectly on your hand specifically. If we try to put it on anybody else, the subtle differences, even if the hands are similar in size, the subtle differences are going to make that glove not fit right. So the same thing here. It has to be the exact substrate that it's looking for. But when it enters that active site, then we have an enzyme kind of modeled out for us and a substrate that will go in. And once it enters the active site, in many instances, that enzyme will then kind of wrap around it and go to work. These are three things that you absolutely need to keep in mind when it comes to enzymes and how they work. All right? Enzymes are very, very much sensitive to changes in pH, changes in temperature, and changes in salinity. Now, salinity is a measure of like salt content, dissolved salt in water. So the more dissolved items you have in your solution with these enzymes, it can affect how well they function or do not function. Right? These are things that we've already mentioned before, that heat and chemical environment affect how a protein can work. Right? But these are the three major ones when it comes to enzymes. If an enzyme is outside of its optimal environment, optimal means like what's best suited for. The shape of this will be changed if it's outside its proper temperature, its proper pH, or salinity, or salty environment. If that site is changed, meaning denatured, then it won't work with the substrate. It will cease to function. And we mentioned this before, mentioning that denaturing of a protein is the same thing as if we were to denature an enzyme. We ruin the shape to the point where it could be ruined to the point where it's not reversible and will no longer function.